The stage was set, but what was still lacking were the scientists with the imagination to see cells for what they really were.
And here in Berlin, one young and ambitious man was about to break the impasse. Theodor Schwann. The two strands of biology, animal and vegetable, were about to come together.
At the time, Berlin was the European centre for anatomy, and the university the magnet for the most brilliant biologists around. Theodor Schwann was keen to make a name for himself and took a position at the prestigious Anatomical Museum. Be warned though, it's not for the faint-hearted.
A guidebook comments, boys will be admitted only in the company of their fathers or teachers, and of the female sex, only midwives will be granted admission. The visitor's attention is called mainly to the wealth of nerve preparations, a long array of monstrous births, and about 500 animal skeletons.
The field of anatomy was in chaos. Nobody really knew what animals or humans were made of. Researchers believed that they were built of many different structures: granules, fibres, tubes, globules, and bladders. And none of them seemed any more important than the others. Animal studies were seriously lagging behind botany, and this was because their cells are so much harder to see. So the scientists didn't really realise there were any cells there at all, and this was fueling the notion that somehow animal tissue was fundamentally different from that of plants. But Schwann used innovative ways to stain his animal tissue. And he had one of the new Lister-style microscopes. He kept finding the same type of globular structure in all the different samples. We know that Schwann was looking at cells, but at the time, researchers used different terms to describe what they were seeing: Kornchen, Kugelchen, and Zellen. The penny hadn't dropped that they were looking at the same thing, and without this connection, they couldn't make the intellectual leap that cells were common to all life forms.
This is a replica of van Leeuwenhoek's microscope. Look how simple it is. It's just a piece of brass, and it's got one tiny hole with a lens in it, which is maybe 1 millimetre across. Yet this is the gadget that transformed the way we see the world.
A tiny lens, yet more powerful than any other. Van Leeuwenhoek knew that it's the curvature of a lens that bends the light passing through it, so making the object being observed, here it's a flea, appear larger. And because Van Leeuwenhoek was a master craftsman, he could curve the lens more than anybody else, almost to the point it was spherical. This allowed him to magnify objects up to 500 times. No one would make a more powerful microscope for over a century. So he now had the technology, but what did he look at?
He went from linen to fleas to the sting of a bumblebee. In fact, pretty much anything he could get his hands on. And one of the things he looked at was water. He'd noticed that in a lake near Delft, the water changed colour with the seasons. And he figured that there might be something in the water that he could discover.
Many of the fundamental breakthroughs of science seem so simple, so absurdly simple. But we shouldn't forget that until Van Leeuwenhoek, no one had the curiosity to find out what might be lurking in the water. He raced home to take a closer look with his microscope. The man who's going to help me study the water is Hans Lonke. He keeps the spirit of van Leeuwenhoek alive by making replicas of his microscopes. To grind a lens, Hans needs no special glass, just a shard from an old jam jar will do. The only way to get enough curvature in the lens is to make it tiny. This was van Leeuwenhoek's secret, and it requires great skill and patience. Van Leeuwenhoek built a staggering 247 microscopes, a new one every couple of months for over 50 years, and told nobody how he made them. We've placed a drop of my lake water onto a slide that slots into the device. With a bit of luck, I'll be able to see what van Leeuwenhoek saw.
Now, I've looked down a fair few microscopes in my time, and this is nothing like anything I've used before. It's fiendishly difficult to use, so I've got Hans with me just in case I can't see a thing. So what do I actually do? Where am I looking?
At the wrong side, you must look through this hole.
Yep.
A very tiny hole, and there's a lens in it.
Yes.
And the lens, through the lens, you see the sample that is put between the glass.
So if I hold it up to the light, like this,
close to your eyes,
and I can see, I can see green.
There is a focusing knob.
There's a focus.
Yes, there is a focusing knob. This might be made.
So I can move it actually away from my eye.
This one. This is the focusing knob.
It's so simple and it works so well. Yeah, that works. That really works. It's now in focus. Oh, wow. Oh my God, you can actually see moving creatures.
Yes.
Oh God, that's incredible. Misha's job here at the castle was to study the chemistry of white blood cells. He decided to look at the large nucleus at the centre of the cell and find out what it was made of. To do this, he needed two things: a ready supply of cells and a way of getting rid of the gloop so he could study the bare nucleus.
Getting hold of white blood cells would normally have been tricky, but Tübingen was the ideal place. The region had been at war with Prussia. Hundreds of injured soldiers with infected wounds lay in the barracks next to the hospital.
Their wounds were oozing copious amounts of pus, which is full of white blood cells. So, revolting as it sounds, Misha collected their old bandages so he could scrape off the pus.
Misha needed something else. His next stop was the local slaughterhouse to collect a pig's stomach.
Oh, look, here it is.
Oh, it's disgusting.
He was interested in the mucus that lined the stomach. This contains an enzyme called pepsin, which helps break down and digest food.
Where's the pepsin?
Look here. This is the pepsin.
Ah, this sort of gloopy stuff.
Yeah.
There's not much of it.
No, that's all.
And this is what helps digest the food in the stomach?
Yeah.
Smells like a pig's stomach to me.
With the pepsin, Misha now had all he needed for his experiment.
And this is a delicacy in Tübingen?
Yes, you can fill it with bread, or you can cut it in stripes, and then you cook it and eat it with bread.
Is it tasty?
Yes.
I'm not sure about that.
Misha carried the bandages and the pig's stomach back to the lab. If he was right, the pepsin would break down the white blood cells. Then, for the very first time, it would be possible to examine the dense nucleus at the heart of the cell.
Nowadays, we do analysis like this using precision equipment. But of course, Misha didn't have any kit like that, so it wasn't easy. First, he had to scrape the pus from the bandages. Now, this is mayonnaise, but you get the idea. Then, he had to wash the pepsin out of the pig's stomach using an acid. and mixed it with the pus. After the enzyme had done its work and digested the cells, only then could he analyse the nucleus on its own.
Now, the big question was, what did the nucleus consist of? Misha spent months analysing its chemistry, and he found it contained a rather strange molecule. This molecule was made up of carbon, hydrogen, oxygen, and nitrogen. He knew these elements were found in all living things. But this molecule contained something extra, phosphorus, and that made it different. Very different.
It was an entirely new kind of molecule. Because he found it in the nucleus, Misha called it nuclein. We now know it as DNA.
Intrigued, Misha repeated his experiment on sperm cells from frogs, carp, bulls, and salmon. Every time he found exactly the same molecule.
Incredibly, some of it has survived. Here in this test tube is some of the first DNA ever isolated. It's DNA from salmon sperm that Misha extracted in the 1870s. Now, it may not look like much, but this brown powder marks the beginning of a scientific revolution. The X-rays taken here at King's College were pivotal. Producing them would be a painstaking job by a brilliant young scientist working with Wilkins, and her name was Rosalind Franklin.
Franklin was an expert in X-ray imaging. With her expertise, the college hoped to be the first to find the structure of DNA. She worked in a new laboratory built in the basement, on the rubble of the old college which had been bombed.
Here in the lab, she took strands of DNA and mounted them inside this specially built camera. The camera chamber was filled with hydrogen to get the very best image. When the X-rays were switched on, they shone through the DNA and scattered in different directions, creating an image on a photographic film.
Franklin took over 100 pictures. Each one could take up to 90 hours of exposure at close range. Once the photo was processed, she projected it onto the wall so she could calculate the exact distance between atoms.
It was picture 51 that showed the best image of the mysterious DNA molecule.
This distinctive X shape was the key that would reveal how DNA is built. But it was not Franklin's name that came to be associated with the discovery of DNA's structure. Wilkins was in close touch with scientists from Cambridge who were anxious to find the structure of DNA before American rivals. Unknown to Franklin, Wilkins gave Photo 51 to James Watson, an ambitious young scientist at Cambridge's Cavendish Laboratory. Having studied the photo, Watson and his collaborator Francis Crick had a sudden revelation. It would transform them into scientific celebrities and put the cell at the centre of world attention.
This is the Eagle pub here in Cambridge. According to Watson, on February the 28th, 1953, Francis Crick strolled into this pub and announced to fellow drinkers, "We have found the secret of life." Now, if you ask me, this story is a little bit apocryphal, probably embellished with some dramatic license. Nevertheless, the sentiment is bang on and shouldn't be understated. This marks one of the truly great moments in the history of science.
Crick and Watson had worked out the structure of DNA, and very soon their double helix model was announced to the world. The structure of DNA is now the most famous image in all biology. What Crick and Watson showed is that it's made of two long strands intertwined into two spirals, with sugar and phosphate making up the backbone. But it's on the inside of the spiral where things get really interesting. On the inside are four molecules: adenine, thymine, cytosine and guanine. They're better known by their letters: A, T, C and G. They're called bases. What Crick and Watson discovered is that these four basic units pair up millions of times within the double helix, and they pair up in a very specific way. A always pairs with T, and C always pairs with G. A and T and C and G making up the rungs of the ladder within the double helix. What Crick and Watson realised is if you split the two strands of the DNA apart, you have all the information to make two new fresh pieces of DNA. Every time you have an A, it pairs up with a T. Every time you have a C, it pairs up with a G. So when you split them apart, you can replace the missing strand and make up a new double helix twice. Genius. This is The Origin of Species, the book in which Charles Darwin outlines the theory of evolution by natural selection. But this copy is unique and totally priceless. This is the first copy of the first edition, the one that the publisher sent to Darwin himself, hot off the press. Now, on page 484, there's a remarkable passage which gives an insight into what Darwin himself thought about the origins of life. All the organic beings which have ever lived on this earth have descended from some one primordial form into which life was first breathed. Now, this doesn't simply say that we humans have evolved from an ape-like ancestor. No, it goes much further than that. It suggested that all living things, from insects to elephants, from a hyacinth to a human, have evolved from one single common ancestor. And it flew in the face of the Bible's account of creation. At the time, Darwin didn't propose a scientific explanation for what created this original life form. His notion that life was breathed into the primordial common ancestor left room for the creator. Yet Darwin's Origin of Species introduced one of the most important thoughts in science: that all life on Earth sprung from one single organism. But to know what that organism would have been, you have to turn to the other big idea around in Darwin's day. It was the theory that cells form the basis of all living things. When it was first proposed in the 1830s, this was a shocking revelation. From man to flowers to frogs, it showed we're all made up of the same building blocks. By the time Darwin published his work on evolution, cell theory was the new bedrock of biology. And crucially, scientists had shown that new cells are born only when existing cells divide. All living cells are descended from other cells. So in just 2 decades in the 19th century, scientists had come up with the two biggest ideas in biology: cell theory and evolution. And if you put the two together, there can only be one conclusion: that all life on Earth began with one single cell. Darwin himself never connected the two theories in such a direct way. But what he did do, just 11 years after publishing The Origin of Species, was make an unholy attempt to explain how life may have begun. He expressed this thought in a private letter to his botanist friend Joseph Hooker. And this time, there was no mention of the creator. And this is it. It's incredible. I've studied Darwin for many years, but this is the first time I've ever seen one of his letters penned by his own hand. And I've got to tell you that his handwriting is appalling. But just listen to what he says. If we could conceive of some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed, ready to undergo still more complex changes. So Darwin had done a complete U-turn. No sign of the creator. He was now suggesting that life on Earth began with chemistry. No wonder he stopped going to church. So how did something as complex as DNA come into existence in the first place? Well, to answer that, some people turned to God, believing that DNA is so exquisite it shows the hand of some kind of intelligent design. Others turned to the stars. And here at Imperial College in London, a surprising discovery has provided a clue to the origins of DNA. At the end of this corridor, pioneers in a field called astrobiology have spent years analysing rocks older than planet Earth itself. These are meteorites, some of which are more than 4.6 billion years old. One of the lead astrobiologists is Dr. Zita Martins. She looks for evidence of life beyond the confines of Earth by searching for particular molecules buried inside rocks from outer space. Hi, Zita. How you doing? So what is it about meteorites that's interesting that can tell us about the origin of life?
We know that specific samples of meteorites, like the ones I have here, they're about 4.6 billion years old. They're as old as the formation of our early solar system.
This is 4.6 billion years old.
Yes.
It's amazing. How
it's exciting to hold these samples in your hands.
And that hasn't changed
No.
in close to 5 billion years?
No.
When you first start looking at this, it looks like a rock, but you are finding stuff in here.
Yes, we're finding organic molecules, extraterrestrial organic molecules that may have been important for the origin of life in our planet, and that's why it's so exciting.
So what you're saying is that up in space, which we think of as a big vacuum, actually there's masses of chemistry just going on, which is actually creating the building blocks for cells.
Exactly. We think the space is an empty place, but it's not. It's bubbling with the chemical reactions.
Right, with activity.
Exactly.
In 2003, Zita got hold of one particular sample whose molecules would reveal something spectacular. It was from a meteorite that had fallen to Earth in 1969, near the Australian town of Murchison. It was dated to be nearly as old as our solar system. Thanks to new technology, Zita's now able to analyse the molecules inside the rock. And she's going to show me why this particular meteorite has made such an impact. Now, this is precious stuff, right?
It is precious. So usually we only have access to milligrams of meteorite sample. So you have to be really careful and not mess up what you're doing.
Yeah, I can feel a sneeze coming on. You might want to hold that away.
I'm going to run away.
It's essential that I don't accidentally ruin the evidence. So, the meteorite is sealed inside a glass tube to prevent contamination from DNA and other molecules on Earth. Once dissolved in solution, the sample can then be analysed by a machine capable of detecting the molecules that make up the genetic code. Okay, Zita, you've analysed the meteorite sample using this pretty awesome machine. Show me what you find.
So what you find is something like this. This represents one of the bases of our genetic code.
So this is a sort of signature for one of the letters that makes up the genetic code for all life.
Exactly.
And this comes out of a 4.5 billion year old meteorite.
Exactly, this is present in the Murchison meteorite.
It must have been mind-blowing to see that for the first time.
Exactly, it was a very exciting and amazing moment, actually.
I mean, conceptually, that's just out of this world. Well, literally out of this
It is
world.
out of this world.
Because what you're talking about is something which is absolutely inherent to life, to every single cell on Earth, and it existed before the Earth even existed.
Mm-hmm. We finally have proven that components of our genetic code were in fact extraterrestrial and were present in a meteorite sample.